Biomolecules. All carbon compounds present in living tissues are called biomolecules. So there are many molecules we know. So among the molecules or those molecules, all those molecules like proteins, amino acids, carbohydrates, nucleic acids are which are found inside the cell are called biomolecules. Now the relative abundance of carbon and hydrogen is higher in cells and lower in our Earth's crust. Okay, so it is an important point, and the table in NCERT that we are going to refer are the comparison of living elements present in living and non-living matter. Among this, you can see that the carbon and hydrogen human body the percentage is much more, whereas in Earth's crust it is very minimal. Okay, now the rest of the molecules, okay, the rest of the elements have almost the same composition both in Earth's crust and human body, but most importantly among these are two: the nitrogen, which is very little in Earth's crust but is abundant in human body, whereas silicon is abundant in Earth's crust but negligible in human body. That's why we can know that many nitrogen and fixing bacteria, cyanobacteria and bacteria are required to fix the atmospheric nitrogen, so that to aid the soil pool of nitrogen, so that the plants can get its enough nitrogen required, because the like human body, the plants will also have a abundant amount of nitrogen, and where this nitrogen came from because of the nitrogen fixing microbes, because nitrogen in Earth's crust is very minimal, and also because of much more competition it gets lot tougher. You will read about this in mineral nutrition. Now we can see that how do we know that which are the molecules and which are biomolecules found in which places. So we can say in our easy way is that we do a chemical analysis. Okay, for plants we take vegetables tissue, for animals we take liver tissue. We take the living tissue and grind it in trichloroacidic acid with using a mortar and pestle, and what we get is a thick slurry. Then we strain this slurry through cheesecloth or cotton. We get two fractions. Okay, the acid soluble or acid insoluble. Among this you have to understand that like if there is a protection in a test tube and we pass on the molecules, then the smaller one will pass and the larger one will remain outside. Similarly, imagine that the solution inside the tube was that of acid, so what we can say that the acid soluble went into and acid insoluble were left out. What is the acid those molecules will be left out, which are the acid insoluble, are known as retentate, and those which went inside are filtrate. So acid soluble filtrate and acid insoluble retentate. We said the smaller molecules will pass into it, so those molecules which have less than 1000 Dalton pass are acid soluble, are found in acid soluble pool or are microbiomolecules. Similarly, those which have more than 1000 Dalton mass, Dalton is a unit of mass, are called above 1000 Dalton of mass are called macromolecules. But generally we said that the macromolecules mass is above 10,000. For micromolecules between 18 to 800 Daltons. So we can say the examples of micromolecules and macromolecules are the micromolecules are amino acids, sugars, nucleotides, and macromolecules are proteins, nucleic acids, polysaccharides. And nucleics are disaccharide, polysaccharide. So the polymeric form, okay. So the polymeric form is the macro, and the monomeric or dimeric or trimeric are micromolecules. Like amino acids is the monomeric form, proteins is the polymeric form, sugars is the dimeric form, polysaccharides is the polymeric form. Similarly, nucleotides, so nucleic acids are polypeptide, are basically many nucleotides arranged. So we can say now that the macromolecules represents the cytoplasmic only the macromolecules of cytoplasm and cell organelle, whereas these micromolecules represent the cytoplasmic composition without the organelle. So the organelle have larger molecules, okay, the biomacromolecules, so the organelles have biomacromolecules. The only the cytoplasmic if we take only the micromolecules, then it is this, then that is the composition of the cell. But the macromolecules of the cytoplasm and cell organelles comes under the macromolecules. So we can say that the acid soluble pool plus acid insoluble pool is nothing but living tissue, because acid insoluble pools give the macromolecules of cytoplasm and cell organelle, and acid soluble pool gives us those of the cytoplasmic composition without organelles. So acid soluble pool plus acid insoluble pool is nothing but living tissue. Now we do S analysis to understand the content of inorganic elements that are present in the cell as well. Now we can see that a leaf or liver we take dried, fully burnt. When we burn a cell or a tissue, what happens? The carbon compounds, the organic compounds, okay, gives carbon dioxide, water, and heat. So what happens is the organic forms are all burnt away. So what we get in the form of S is the inorganic forms, the calcium ions, magnesium ion, sulfates and phosphates. Okay, so +6 oxidation is sulfates, is +5 oxidation phosphates. So now we will refer to the tables associated in the NCERT. But first let's have a look at the all the theory part in this in the first two sections. First, primary metabolites that are required for basic or primary processes like photosynthesis, respiration, protein plus lipid synthesis. These are the main fundamental processes that take place, and for these fundamental processes we do need the producers need these processes to sustain itself. It needs to function these processes, and those molecules which are required to run this process are called primary metabolites. Now there are some which are not used to run these processes. These are called secondary metabolites. Listen carefully. The NCERT states that secondary metabolites seems to have no direct function. It doesn't say that there are no functions. It says this seems to have no direct function in growth and development of its producers like plants, fungi, microbes. It do not have its direct functions. Then what is the needs of its having? Honestly, that we humans are extracting usefulness and viability from these secondary metabolites like flavonoids, drugs. We'll see the examples next. Like the primary metabolites are identifiable functions and play a role in normal physiological processes like the aqueous sugars, amino acids, sugars, chlorophylls, hemes. Similarly, the secondary metabolites have like a role in host is not understood. Okay, role in host is not understood, but humans have very good use of this substance like rubber, drugs, species, sands, pigments. Now let's talk about some important points among this chapter. In this chapter basically we deals about the macrobiomolecules, these proteins, polysaccharides, nucleic acids, lipids. Lipids are not strictly macromolecules. Okay, among these four biomolecules we can say that a proteins is a polymeric form, polysaccharide is a polymeric form, nucleic acid is a polymeric form, but lipids are not polymeric form, it is the monomeric form. So lipids are not strictly macromolecules, molecular weight do not exceed 800 daltons. Then why do we study them in macromolecules? It is because they come under the macrobiomolecules because they are present in membranes. Okay, we know that in membranes, the bilamellar model, we know there are hydrophilic heads and hydrophobic tails. So these because these hydrophobic tails, because of the presence of these hydrophobic tails, when we grind it, okay, on grinding they form vesicles. Vesicles means circles. Okay, these are called vesicles, means just spherical objects. They form spherical bodies they are formed, and because of this hydrophobic tail they are aqueous insoluble. So where are they soluble? And they acid soluble. So they are they are acid insoluble. They are aqueous insoluble as well as they are acid insoluble. So we can say just like here that the molecular mass of lipids does not exceed 800 daltons, but still they are considered as acid insoluble, because our first criteria between macromolecules and micromolecules is the acid solubility and acid insolubility. The weight comes in the second criteria. All those which are acid insoluble are called macromolecules. That's why lipids are macro biomolecules, but are actually sized in the range of micromolecules. Now we will see a few tables in NCERT. We have studied the first table. Now second table, look, you can see the inorganic ions. All the inorganic elements exist in the form of ions. Okay, so we can say that sodium, potassium, calcium, magnesium, water compounds are important substances. Among this, you can see that there is a question was asked in 2013 NEET, that which is the most abundant intracellular cation. Then the answer is K plus. Okay, K plus is the most abundant intracellular cation. You will learn about this in zoology part. Okay, now the examples of secondary metabolites. We have seen alkaloids, flavonoids, rubber, essential oils, antibiotics, colored pigments, sands, gums, spices. Now let's see this table, third table of the NCERT. It questions comes in exam for matching these contents. Among this, you can see that pigments, we not carotenoids, anthocyanins. I told you that the secondary metabolites seems to have no direct roles in the hosts. That doesn't mean it do not have any role. It is limited because of our understanding of biomolecules chapter. But if we have learned the chapter on photosynthesis, we do know that carotenoids are light harvesting complexes which broadens the spectrum of light that it can initiates the light reaction. So carotenoids is a important molecule, and so then anthocyanins. But because of our limited understanding in this chapter, we'll consider this as secondary metabolites, carotenoids, anthocyanins. Now among the terpenes, you can have terpenoids, terpenes, monoterpenes, diterpenes. It is easier to remember. Essential oil, lemongrass oil, it is easier to remember. Now among the polymeric substances, we know rubber, gum, cellulose are polymeric substances. And after those, we have to learn them or memorize are the morphine and codeine are alkaloids. The abdomen, ricin are toxins. Okay, we know that toxins have smaller names. Okay, the toxins. Lectins have concanavalin A, and drugs are vinblastine and curcumin. One more important table that we have is this one, that the water content is water is the most abundant molecule in any cell. Okay, any cell, water 70 to 90%, and then the ions is no more than 1%. Among the macromolecules, we can see that the proteins is the most abundant one, 10 to 15%, and others are in a small amount. Among this, we have to understand that with the water. Okay, so with the water, you can see the 70-90%. If in a cell there is only water, it means that the wet weight, okay, wet weight is very greater than dry weight. Okay, when we dry a tissue, then what we get is its dry weight. Before drying, with the water content present in it, what we get is wet weight. So that's all. Further, that there were some questions asked in our entrance exam. Among this, it is important question is that about bioinformatics. What is bioinformatics? Bioinformatics is nothing but collecting storage and analysis of large amounts of biological data in computer to make useful conclusions. Okay, so this is the bioinformatics. Now there is one more question that was important. Correct order of abundance of oxygen, sulfur, magnesium, calcium in the earth's crust is referred to the table we just saw in NCERT. Among this, we can see that anyone who knows that oxygen is most abundant in the earth crust, even in the human body, so he can take the correct answer. Oxygen is greater than calcium, is greater than magnesium, is greater than sulfur. So that's all for today's class. Thank you. Signing off.